speaker. It's my pleasure to introduce our next speaker for today's Epilepsy Connect Symposium. Our speaker for the topic of the genetics of epilepsy is Dr. Kristen Park. Dr. Park is an associate professor of pediatrics and neurology at the Children's Hospital of Colorado, specializing in epilepsy and neurogenetics. Dr. Park graduated from the Mayo Medical School and completed her neurology, her neurology and pediatrics residencies at the Children's Hospital of Philadelphia, followed by a neurophysiology fellowship at the Children's Memorial Hospital in Chicago. She currently serves as the Associate Director of the Epilepsy Fellowship Program and the co-director of the Neurogenetics Clinic here at Children's Hospital. She's participated in a number of team science efforts, including the Epilepsy Phenome Genome Project, the Epilepsy Genetics Initiative, ClinGen, and the Human Epilepsy Project, which have yielded important discoveries about the genetic underpinnings of epilepsy. Her current research efforts include studying the natural history of GRIN disorders, collaborating on a review of PTEN-related brain malformations, and examining the role of genetic mosaicism in resected brain tissue from epilepsy patients. Dr. Park is also on the board of directors of the Epilepsy Foundation of Colorado, Wyoming, and is a regular physician at the Epilepsy Foundation summer camps. She's an amazing volunteer and friend of the Epilepsy Foundation, and we're delighted to have her as one of our speakers today. Dr. Park, I'll turn it over to you.
Thanks so much, Sarah. Let me pull up my slides here. And do I let's see, share screen? All right, can everyone see that?
Yeah.
Yes, okay, great. All right, so I'm super excited to talk about epilepsy genetics today. This is one of my favorite topics, as you may have gathered, and I think something that has really been groundbreaking since I started practice about 13 years ago. So disclosures, I am a site PI for an upcoming clinical trial involving a therapy for genetic epilepsy. So the outline for today, we're going to talk about an overview of genetics. I want to make sure everyone kind of remembers their high school biology and is using the same terms to mean the same things so that we can understand what we're all talking about. We'll talk about genetic discovery in epilepsy that has taken place over the last 10 to 15 years and the current state of epilepsy genetics. And then we'll also spend a little bit of time at the end talking about some novel applications and future directions of where this type of research is leading for patients with epilepsy. So let's go back to the basics. If we talk about our body, it's composed of some complex molecules, lipids or fats, proteins, and carbohydrates or sugars. And each of those molecules has a very specific role to play in a part of the body. They make up our muscle fibers, our cell membranes, our hormones, our antibodies, our skeleton, all of those things. Typically, lipids and fat are in membranes, and proteins are often our enzymes and important body, kind of mechanism providing parts of our makeup. And antibodies are the same, they're also proteins. So I'd like to kind of sum everything up with this video that I found that kind of goes over what's called the central dogma of genetics, how our DNA gets translated and turned into proteins. So I tried this earlier and it does work. So I'm going to see, it did work. Let's see. Wait, maybe. Here we go. Okay. Can everyone hear that?
Very quiet.
See if I can, I'm at mix. Okay. Sorry. Okay. So, hold on. Now we're just playing it again. Hold on. Okay. Can everyone see my slide again? Where we are. I'm sorry that the video had low volume. So I think the take-home points from this are that our genetic codes are recipes for how to make us as human beings. Again, we start with our DNA, which gets translated to RNA, which gets translated to proteins. That messenger RNA has to be spliced. That's the term for cutting out those portions that we don't need. Those 3-letter sequences that make an amino acid that I've listed here are what we call codons, and that is kind of the word as it's read by our ribosome. There is a fair amount of redundancy, so there's only 20 some amino acids and a lot more combinations of those 3-letter codes. And the reading frame is very important. If you imagine reading a book and you shifted all of the letters over one, what you read after that shift would probably be kind of nonsense. And so that is very important as the DNA is being translated. I think of this splicing, I put this little picture of a train up here. I think of our genetic code like a train. It has these boxcars that contain important information called exons, and then those linkers in between the boxcars that get cut out in the splicing, and those are called introns, and we'll hear about them later. Most of our genetic transcription is quite accurate, but occasionally typos occur and different letters are substituted at different places. And that's important because that's what allows for the breadth of human variation that we see between members of the human race. And it allows for evolution to occur and people to adapt and change over time. So additional definitions to cover before we start. So autosome, we all have 22 pairs of human chromosomes aside from our sex chromosomes, and those are called autosomes. Our sex chromosomes are inherited differently, right? So two males have an X and a Y, and females have an X and an X. And therefore, males are responsible for determination of genetic sex by passing on either their X or Y chromosome to their children. The genotype is kind of the content of someone's genome. Those letters in their spelling is the genotype. Phenotype, as a word, means observable characteristics of an individual. That is produced by interactions between our genes and our environment to produce this physical expression of those genes. We're going to talk a lot about what's called Mendelian inheritance. I put a little thing down here on the side. This is Gregor Mendel. You may remember him from your biology. He did the breeding of the peas to give them different characteristics, and he was a monk. And so we talk about Mendelian genetics to mean an inheritance pattern that follows those laws that he outlined. And that means that it's usually dealing with one gene, and it is inherent from either parent at an equal frequency. Allele is the gene spelling that you get from your parent, and sometimes they are the same and sometimes they are different. When we talk about conditions, we will often talk about conditions as being either autosomal recessive or autosomal dominant. Recessive means that you have two copies of each gene, right? One from your mom, one from your dad. And in order to manifest the condition, you have to have both of those copies not working. If you have only one copy not working, you're considered to be a carrier of that condition. On the other hand, in autosomal dominant conditions, if you have both copies and one is not working, you will express the symptoms of that condition. As opposed to diseases that are on our sex chromosomes. Most of them are called X-linked, which is more likely to affect boys because they only have a single X chromosome, as opposed to women who have two X chromosomes and kind of have a spare if one of them is not working. Most of the time, X-linked conditions are passed from unaffected mothers, again, who don't have any problems, to boys. And that's the same for hemophilia, which is a well-known X-linked condition from Rasputin and the royal family in Russia. Mendelian genetics also relies on something called a Punnett square, which you also may remember from high school biology. So we have on one side the genes that you're going to get from your mom, and on the top the genes that you're going to get from your dad. They combine and they end up in these different combinations. Dominant genes are expressed with a capital letter, and recessive alleles are expressed with a lowercase letter. If you carry the two dominant alleles, you're here. If you carry one of each in either combination, you're considered to be heterozygous, meaning that those genes are different in their expression, and you have one copy of each. Whereas if you carry two copies for a recessive condition, you're called homozygous. And those are just words that we're going to use in the rest of the talk. And this is kind of what we as geneticists use to help predict the frequency that diseases may run in families. So if these people were real people and not just letter Bs, they would have a recurrence risk when talking about having additional children. And recurrence risk typically for recessive conditions is 25% for each pregnancy, but the risk of having a carrier, again, who doesn't have any symptoms themselves, but could pass it along to their child, is 50%. For a little bit of perspective, all of us carry about 20 recessive disease alleles for various conditions: cystic fibrosis, hemophilia, any number of different things. And that doesn't really cause us any problems. That's just part of who we are. Problems happen when we meet up with someone who also carries the same risk condition, and then we would have an affected child. In another piece of perspective, there are about 3 million different spellings between any two people on the planet. And again, those allow us to be different and have different characteristics, different hair colors, different responses to disease, different likes and dislikes, and all sorts of things of that nature. So if we're going to translate some of that general genetics knowledge into epilepsy, how does that work? So we've known that epilepsy has a genetic component all the way since Hippocrates in 400 BCE. He made the observation that epilepsy often ran in families, knew that it was attributed to the brain, and kind of said, this is what I think about it. And it's taken us a long, long time to figure out what the actual underpinnings of epilepsy in the genetic world are. The first studies were done, back in from around 1930 to 1990 in large populations, and indicated that if we think of the general risk of epilepsy in the population as about 1%, meaning 1 in 100 people, the risk of epilepsy in someone who has an affected parent is about twice that, so somewhere between 2 and 4%. So higher than the general population, but not to the point of a single gene. Remember, we said if it's a single gene risk, the risks are going to be about 25 to 50%. So this is lower than that, but it's still there. If multiple family members are affected, then the risk can increase, and that suggests potentially a stronger genetic component than what we found in these population studies. Sometimes that's based on syndrome. So they found that patients with absence seizures had a higher risk of genetic inheritance than those with other types of epilepsy. And we also know that even when we think of epilepsy as an acquired condition, so somebody who's had a brain injury or a stroke or some other thing that they weren't born with, the risk of developing epilepsy after those types of incidents is actually somewhat genetically mediated. So they've done studies in patients who have had alcohol withdrawal induced seizures, and they found that they have a higher incidence of relatives with epilepsy than people who did not have alcohol withdrawal related seizures. In children with epilepsy that develops after a stroke, those children more often have a family history of epilepsy than those who do not develop epilepsy after a stroke. So there's still a very powerful component there, even when the whole epilepsy itself is not genetic. So this is kind of the outline that I'm going to kind of use to talk about a little bit today. So if we think of epilepsy in general and divide it again into genetic and acquired, that genetic component is either Mendelian, meaning dealing with one gene, and that's most of what we know now about epilepsy genetics. Is epilepsy genetics determined by 1 or 2 at the most, typically one gene. But a lot of epilepsy genetics is probably polygenic, meaning that it's determined by a number of different variations in how our brain genes are working, and all of those add together to give people a risk for epilepsy. Again, we talked the fact that there might be some subtle genetic influences in acquired epilepsy, and we're not going to really talk about that very much today because that research is pretty much in its infancy. So if we talk about Mendelian genetics, I tell patients that when I started practicing about 13 years ago, we probably could test for 1 epilepsy gene. We knew about 1, maybe 2, and we didn't have the ability to test for many of them. But since that time, the field of epilepsy genetics has exploded. And currently there are about 800 genes that we know primarily cause epilepsy as their main manifestation. And there are about 3,000 more that are associated with changes in how someone develops and may have epilepsy as a symptom, in addition to other things like intellectual disability or autism or movement problems. Most of the time, these Mendelian epilepsy disorders are de novo, and that term means that it occurs only in the patient. So they typically don't have a family history, they don't have several other people and a cousin and a grandfather with epilepsy. It's only that child or adult that has been affected. There can be, we now know that about 10% of parents of those children do have something called mosaicism. And mosaicism is when not all of the genes in your body are spelled the same way in every cell. So we test either cheek cells or blood cells for our tests of epilepsy genes. And that's not every cell in your body. And so there may be other cells, your egg cells, your muscle cells, your eye cells, that have a different spelling of the gene. And that's important because if that's the one that gets passed along to your child, then you may have a risk of having another affected child. And so that's really important. There are other types of genes that can be passed on, and the family history of epilepsy is not known because of something called incomplete penetrance, meaning that the child may manifest the symptoms of epilepsy, but the parent who even has the same spelling of their gene didn't end up manifesting any symptoms of epilepsy. Perhaps they didn't come in contact with the same environmental issues, or they didn't have the same combination of genes that work together, and so they never had any symptoms. There are, of course, familial epilepsies, and we do know more about those. Some of the genes that are listed here are seen in families. And, you know, as somebody who works in epilepsy genetics, I frequently am dealing with pediatric patients, but there is additional literature that says that if we were testing adults on a more regular basis, the yield could be up to about 40%, depending on who we're testing and what type of testing we're doing. So this is something that I am in contact with frequently, but is actually relevant for adults with epilepsy. So let's have an example about these epilepsy genes and how we discovered them and how they may be impacting treatment and management of epilepsy. So there is a well-known, this is one of the first epilepsy genes, and it is related to Dravet syndrome. And the gene is SCN1A. All the genes are named by letters and numbers, which makes them kind of confusing. We call it alphabet soup, but this stands for sodium channel 1A. So it's a member of a family of genes that are responsible for the movement of sodium currents in and out of our cells, especially our brain cells. So this clinical presentation, Charlotte Dravet was a French epileptologist who noticed this group of patients that all kind of behaved the same back in 1978. They had seizures that were often induced by fever. They lasted for a long time. They involved one side of the body. They subsequently developed myoclonic epilepsy. And she said, I wonder if these children all have the same thing. And it wasn't until 1997 that some researchers in Australia looked at a very large family where the core of that family had about 25 people who had epilepsy over 4 generations. And members of that family had different types of epilepsy. So sometimes they maybe just had febrile seizures. Sometimes they had epilepsy that was intractable. Sometimes they had generalized seizures. Sometimes they had focal seizures. They were all a little bit different, but they all had epilepsy over this 4 generations. And so they did some of the technology that they had available to them at the time called linkage analysis. And they said, hey, we found that all of these people in this family have a variation or a mutation in the spelling of their SCN1A gene. And that was in 2000, so several years after that. And so then they said, well, these patients had febrile seizures, and these patients with this Dravet syndrome have epilepsy that's often provoked by fever. I wonder if they have the same thing. And they do. In fact, they all have variations in their SCN1A gene. And so now this is probably the most common epilepsy gene that is tested for and found in our genetic testing era. And what has happened since 2000 that allows us to move the field forward and translate this into other therapies? So now there are huge databases about how the gene SCN1A is spelled to help us interpret these test results across the whole world. We now have information about which medications are most effective for children with this particular genetic conditions. And we're developing new targeted medications based on this genetic discovery and being able to use animal models and test a variety of compounds on this type of epilepsy. Some of the changes in spelling of SCN1A have also been implicated in epilepsy in general. So these children are obviously very severely affected and have a rare epilepsy syndrome, but SCN1A as a gene has actually been implicated in more common epilepsies. And so this information may translate to the broader field of patients with epilepsy in general. And then most excitingly, we are now developing gene therapy for patients with Dravet syndrome. And we'll talk about that further along in the discussion, but that's really exciting. So why does it matter to have a genetic diagnosis for epilepsy? Well, we obviously talked about there can be some implications for recurrence risk for both the patient affected by epilepsy and other members of their family. We can give more accurate prognostic information about what we anticipate may happen with their epilepsy over time. We may need to do additional monitoring. So some of these other conditions are associated with changes in someone's heart or breathing or kidneys. And if we don't know that those things need to be monitored, we wouldn't order those tests generally. When they've done large studies of patients who've done genetic testing for their epilepsy, about 40% of them have a change in how their epilepsy is managed based on their genetic diagnosis. And then I think the last part of this is something that I talk with families a lot about, something called a diagnostic odyssey, where many children who are affected by epilepsy, the families have asked why for quite some time, and they've never gotten answers. And before we had all this genetic testing, they often subjected their child to many low yield, meaning it was unlikely to give them an answer. It was expensive, it was invasive, they had spinal taps, they had muscle biopsies, they had surgeries and all sorts of procedures to try and get a list of all these potential diagnoses down to something that was the reason for their child. And that was often very expensive and took a long, long time. Most of the time, children are those who are affected by these rare diseases. And before genetic testing, it would take an average of 5 years before they got a diagnosis for why their child had started having seizures. And I put some quotes from families there. You know, they express feelings of limbo. They have a comfort from finding an answer, and they have again information about early awareness and screening that helps them to prepare for the future. So that's our overview of Mendelian epilepsy genetics. I'm going to talk now about non-Mendelian epilepsy genetics. Again, this is where one or more genes are combining together in their effect to produce the symptoms. And that's what's on display here with these graphs. So if we have one gene, it's the top graph there. If it's more than one gene, you can see it starts to resemble this normal curve that's on the right. And the concept is that people have a combination of lots of different variations in how their genes are spelled, their brain genes. And at a certain point, if all of those add up together, like a seizure threshold, past that point, they will begin to be affected by those changes and manifest epilepsy. So how often does that happen? And we suspect that when we've looked at large groups of patients in epilepsy clinics, typically about 12% of them have someone in their family who has epilepsy. It's typically not a parent or a sibling, a first-degree relative. It's typically someone more distant, a grandparent, a cousin, you know, mom's first, you know, cousin once removed or what have you. Again, suggesting that it is more complex than just a single gene and a pattern that you can trace through the family. This type of complex inheritance is what we think is at work for things like height, intelligence, weight, other things that are not predicted by one gene and a simple little square that we saw before. And so, again, this is kind of just explaining what I showed pictorially, that this risk, that these different spellings attribute risk to people that kind of are cumulative until they get to a certain point after which the symptoms, in this case, seizures, are manifest. So, this is pretty complicated to figure out if we think about it. And it requires a large number of people with epilepsy to get this type of information. And the reason is that if we think about our Mendelian epilepsies, it's one gene, and the results of a problem in that one gene are significant enough to cause someone to have epilepsy, which is diagnosed and manifested. But if we're talking about lots and lots and lots of genes that have to add up together, each of those genes probably only has a small effect on whether someone has seizures. And how do we tell whether it's going to be a problem or whether it's just part of human variation? And in order to do that, we have to have hundreds of thousands of people together to say, how often do these types of changes show up in people with seizures versus how often do they show up as just part of human variation? And so the papers that I've quoted here from, let's see, this was the Epi25 collaborative, which I was involved in, and then the ILEE, the International League Against Epilepsy Consortium on Complex Epilepsies. So these studies gathered large groups of patients with epilepsy and tried to compare them to people without epilepsy. And the first one showed that when they had 9,000 cases and compared those 9,000 cases to probably double the number of patients without epilepsy, none of the genes that we know about rose to statistical significance. So we had 9,000 people with epilepsy who had agreed to participate in research, and we didn't find any answers. That's how complex it is. There were some that were pretty close. They were pretty close to being statistically significant. And it's interesting to note that these genes are some of the same genes that are involved in those rare and severe epilepsies in children. So indicating that there's a large overlap between the genes that are involved in kind of garden variety epilepsy that is seen at many clinics, and those children with very, very rare epilepsies that we think are mediated by one gene alone. And so that's important for our mechanism and our treatment discussion. The other study published in 2018 doubled the number of patients with epilepsy, so now, or almost doubled, so now they're at 15,000 cases and almost 30,000 controls. And they did actually find some genes that were statistically significant. And again, there was significant overlap with genes that we already know are associated with epilepsy. There is another study that's underway that is actually, again, doubling the number of patients with epilepsy that are being studied. So 30,000 cases, that's about the same number that other similar studies use for studying things like schizophrenia, which is also thought to be polygenic. And so we hope that those results will be coming out soon. I said anticipated in 2021, they're not out yet. So it will be very interesting to see what they discover. So the goal for this is to hopefully, and this is emerging research, so is to hopefully come up with something like what they've started to do. I think this was out of Ohio, which is something called a polygenic risk score. Can we sequence someone's DNA and then give them a score that says, this is how likely it is that your epilepsy is genetic, and this is how likely it might be that you have a risk of passing that on to your children. And I think they have been able to do that for large groups of people. They need to sort those people by ethnicity because obviously we come from different places and our genes are different. And they've been analyzing thousands of variants. Sometimes there's a little bit of difference between generalized epilepsy versus focal epilepsy, but not always. And certainly this is really emerging research. So it's nothing that's going to be in the clinic now where you can come in and say, what is my risk score for having another seizure after an unprovoked seizure? What is my risk score of having a child affected by epilepsy? So this is where we would like to be, but not yet where we are. Another area of research is in structural epilepsy. And so Dr. Ma talked about this a little bit. We often see epilepsy as arising from one single spot in the brain that may have not formed correctly. And there are lots of different pathological distinctions between those areas. Sometimes it's extra folds in the brain, sometimes it's enlarged cells that did not migrate properly, sometimes the brain is overly smooth or overly bumpy, or it's enlarged in one area. Those are the terms that I've listed there. But the most common cause is something called focal cortical dysplasia, and that's often visible on MRI, but not always. And this type of lesion on the brain can frequently lead to very treatment-resistant seizures and often progresses to surgery. And so what they actually did to bring this field forward was to look at surgical specimens from patients who had undergone epilepsy surgery, and they identified what we called a somatic mutation or evidence of mosaicism. And that's what this picture here on the right-hand side is showing. So this is mom and dad. They've had their egg and their sperm joined to form the baby, and then the baby has to, the baby's initial cells have to divide over and over and over and over again to produce the millions of cells that make up a human being. And at some point in that process, something happened, and one of those genes got transcribed incorrectly, and it made a mutation. And it only occurred in the cells that were destined to become the brain, not any other cells in the body, just some of the ones that were destined to become the brain. And so when that happens, you may see that those cells subsequently divide to make more and more brain cells, and only some of the brain cells may have the mutation while others do not. And that may lead to this structural epilepsy and this focal dysplasia where the connections didn't happen correctly, and that area generates seizures. And so they studied about 300 specimens and identified a mutation of that type in about 13% of the cases. Typically, those mutations were in a very similar pathway called the mTOR pathway, which is a very important pathway in our body that is responsible for how cells divide and grow. And importantly, they also found that there was a gradient of the percentage of those mutated cells. So when they monitored those patients with epilepsy, the place where the seizures started had like 50% abnormal cells, whereas if they went out further and further, they saw maybe 30% and then 10% and then 5% at the border. And I think that's important because what if we could determine how much tissue to resect in a surgical specimen by how frequently those mutations were seen? So if we did the surgery, we took out the 50% area and then we analyzed it again, and it was still at 40 to 50%, we would keep going and we would take out more until we got to maybe 10% or less of the cells had that same mutation. This is something that's done in dermatology already. It's a surgery called Mohs surgery, where they take out a cancerous lesion on the skin. They examine it under the microscope. They say, no, there's still some cancerous cells there. We better take a little bigger area. And then we're going to examine it again under the microscope. And then we're going to take a bigger area, right, to guide that surgery in a way that's clinically meaningful. So I think the other important takeaway from this type of research is also how are we testing, right? So if we're testing patients for epilepsy genes and we're testing their blood, are we missing something? Should we be testing something different? So if we think about how people embryologically develop, our blood turns into things like our muscles and our skin. They're derived from the same types of cells, but our brain comes from things like our cheek, our eyes, our spinal fluid. All of those things are the same types of cells. So what if we don't take blood to analyze? If we're talking about epilepsy, what if we took some cheek swabs, or we went into the nose, which is kind of connected to the brain, and we took some cells from up in here, or we took spinal fluid? We'd be able to get better answers for patients by testing the tissues that are actually involved in the disease. This is also something that's exciting for targeted treatment. So we talked about mTOR, that pathway, and there are already medications that exist that are have been developed and tested by the FDA and are used in cancer care on a regular basis. And can we then, if we know that someone has epilepsy because this gene is mutated in some of their brain cells, could we use those medications and treat their epilepsy better? And that actually has been explored a little bit in tuberous sclerosis, which involves this same mTOR pathway, and some of the mTOR inhibitors, including everolimus, which people may have heard of, have been used to treat tuberous sclerosis. And so could we do that on a larger scale for patients with other types of structural epilepsy? So the next thing to talk about is emerging research in gene therapy. So we talked about, we have these epilepsy genes, and sometimes we have people who have epilepsy or have variations in those genes that is the source of their epilepsy. Could we make something to use that and have it become a treatment? And the most studied application that we have now is something called antisense oligonucleotides, or ASOs. These are short strands of connected nucleotides that can alter that RNA and reduce expression of abnormal genes. So typically they are again short, between 8 and 20 nucleotides. They bind to that RNA and they say, don't actually make this part of the gene because it's not working correctly. And this technology has been around since about 1998. The first ASO was approved for the treatment of a very severe type of retinitis that occurs with cytomegalovirus infection. And unfortunately, since it occurs in the eye and it has to be treated locally, it did involve an injection into the eye of this compound. But since then, we've gotten a little bit better. And a couple of ASOs were recently approved by the FDA in 2016, both of them for a very severe degenerative muscle disease called spinal muscular atrophy. And they are now in widespread use, including in our hospital. And most of these have been developed for, and there's another one being developed for NCL, which is also a very rare and progressive degenerative disease. And so most of these ASOs have been approved thus far for progressive diseases because the technology itself is preventing additional abnormal copies of that protein from being made, but it is not able to go back and undo some of the things that have already happened in the development of the brain or in progression of the disease already. So we're going back to SCN1A now, Dravet syndrome, which we talked about earlier. And this is one of the treatments that is being, one of the gene therapies that's being developed for Dravet syndrome. It's called TANGO, target augmentation of nuclear gene output. So this is the graphic from Stoke Pharmaceuticals, which is the sponsoring company that's doing the initial studies. And so you can see what's happening here is this is our DNA. And it's normal DNA. It is made and then spliced and then turned into productive or translated into a protein that exists. And even in the normal gene, there is something called an NMD exon, or nonsense medium decay exon. And this is an exon that causes the body to say, oh, don't make any more of that. We're gonna, if we put that in there, it's gonna cause nothing to happen. And so you lose about half of your protein on a regular basis just because this exists in the body. But if you already don't have enough SCN1A because of a gene mutation, what if we could get that to actually produce functional protein and make up for some of that loss? And that's what this therapy is designed to do. It blocks the inclusion of that exon and allows for additional RNA to be translated into protein. This Medication is delivered by intrathecal, so into the spinal fluid, into the by spinal tap, and has been studied only so far in mouse models, and it helped to decrease mortality from this condition and seizures. The clinical trials for this began in 2020. No results have been released yet, but I think it's very promising. Other types of gene therapy are also being investigated. So this is kind of something that kind of binds on to a normal process that's already existing in the body. But there are types of gene therapy that are being developed to transfer functional copies of genes into people or edit copies of genes that people already have to prevent diseases from having severe symptoms. And so a lot of these trials are already underway for other neurogenetic conditions that have epilepsy as a main symptom. And those include Rett syndrome, Angelman syndrome, Fragile X, which is also one of the leading causes of mental retardation in young boys and men, SCN8A, so that's another sodium channel in a different family of sodium channels, but also related to epilepsy. And as I mentioned, tuberous sclerosis is also having some gene therapy trials as well. I think if you look online about any of these things, you need to be pretty cautious. There are many researchers for things like Parkinson's and ALS that say, you know, we've cured those diseases about 20 times in the mouse, and we have cured them in humans, not yet. And so, you know, there is a lot to translate a therapy from a mouse to a person, and it doesn't always work. And so we have to interpret these with caution. And there are a lot of challenges that still exist before we can use some of these gene therapies. We have to make sure they don't edit other genes that are functional, that we get them to the right place in the right dose, that our immune system doesn't attack them and then, you know, does allow them not to work. So there are a lot of different challenges that still need to be overcome, but I think this is very exciting work. So in conclusion, what I would say is that there is a lot of evidence that suggests that epilepsy has a strong genetic component, and that exists in many types of epilepsy, even without a family history, and epilepsy that even at first blush may seem to be related to a particular incident or an acquired cause. Most of the genes that we know about have been discovered in relation to severe pediatric epilepsy syndromes that often have a number of other neurologic conditions associated with them. Typically, those are related to one gene or Mendelian inheritance. However, we are translating that knowledge also to less rare epilepsies and into adults as well. We need cooperation in order to continue genetic discovery for more common epilepsies and generalized epilepsy syndromes that I've listed there, you know, and that needs to be on the order of thousands of people participating in research. I think genetic diagnoses can affect our treatment decisions. We saw up to 40% of patients have changes made based on genetic diagnoses. And we are developing new exciting therapies that are part of an overall effort in medicine called precision medicine, meaning getting the right therapy to the right person in the right dose, and addressing the underlying root cause of the medical condition rather than, you know, seizure medications, which are you know, more of a suppressant of seizures and not really addressing actual causes of epilepsy. All right, so that was a lot to take in. So I will stop sharing. I don't know, I can't see the time, so I don't know. Oh, I had 1 minute, so I don't know, Sarah, if there are questions in the chat, if you have been monitoring that, if you wanted to.
Yeah, we do have a couple of questions. Thank you so much, Dr. Park. That was such a comprehensive overview. Thank you. I really appreciate how you made a really complicated topic pretty accessible. I mean, I think it was still complicated, but you covered a lot of really great information. And, you know, I think a lot of that is material that's not generally accessible to the general public. So thank you so much for that really, really fascinating presentation. There are a couple of questions that we have in the chat. So I'll start with those. The first one is genetic testing readily available and affordable for most people?
Yeah, so that's a good question, and that's a complicated issue. So I think that is a reflection of, you know, kind of our healthcare system in the United States, that there are some insurance companies that will cover genetic testing, but not all of them do. And mostly they think of it as still experimental, even though there have been a number of studies that have shown that it can affect treatment. It's typically authorized for children, and it's less common for adults. There are companies that have self-pay programs, and the cost of genetic testing has come down quite a bit. For our patients, there are actually several no-cost genetic testing programs for children under 8 who present with epilepsy that are being sponsored by some of the pharmaceutical companies and family organizations dedicated to research in these genetic epilepsies. And that's been a development maybe over the last 5 years or so. And so we typically do those routinely at Children's for patients that are eligible to get them genetic testing.
Great. If we have no answer on why our son was diagnosed with epilepsy at age 26, would it be beneficial to be genetic tested? And I'm not sure if that question is to have the son have genetic testing or to have the family.
So people think of genetic epilepsies as also encompassing those that occur under age 40. So if we think about epilepsy, the onset peaks are in children less than 2 and the elderly. And when we did some overall studies of genetic epilepsies, we included individuals with epilepsy onset up to 40. And so I think 26 is still worthwhile to have genetic testing. I think the yield is potentially lower because, as I said, most of the genes that we know about are ones that cause the severe epilepsies in childhood. So when you get to somebody who's had a healthy life for 26 years and then develops epilepsy, the effect size of that gene is probably smaller and more difficult to pick out from the background noise. And so the chances that the testing is going to be unrevealing are probably higher. And I guess still, I would say still worthwhile.
And a similar question, an adult said, I have epilepsy. Would it be worthwhile to ask my relatives to share their genetic tests with me?
Most definitely. If they have had testing for, for epilepsy or other conditions, I think that could be informative. Sometimes I think epilepsy in the family history is hard for us to get because of stigma. And so people say, I don't know anything about my mom's epilepsy, or I don't know anything about my grandfather's epilepsy because they never talked about it. And so it can be complicated, but now we have the ability to access that without needing a lot of information.
Right. Another question, my granddaughter has an unbalanced translocation on 5 and 6. Would gene therapy apply to this at all? It came from my daughter and then from myself.
Yeah. So, epilepsy genetics or epilepsy is a manifestation of a number of different genetic conditions. So single genes, but also if we think about what are called microdeletion and microduplication syndromes, that's where a whole chunk of genetic material is either lost or gained, right? Deleted or duplicated. And if you think about it, if there's this much genetic material in that we're talking about, one of the genes in there probably has something to do with how the brain works. And so when you have microdeletion and duplication syndromes, often epilepsy or other neurologic conditions are part of the symptoms. The critical question is, which gene is it in that region? And so sometimes research needs to be done to say, if we compare a whole lot of children who have deletions or duplications in the same area, what is the overlap? If one child has this and the other child has this and another child has this part and another child has this part, what gene do they all share that might be related to epilepsy? And if that can be identified, then you can move forward and and say, how could we affect that gene and mitigate the symptoms of epilepsy? I think the complicating part is that you may not be able to change some of the other genes that are involved as well. There's obviously more than just that one gene related to the epilepsy that's included in that area. And so, the gene therapy for that is much more complicated. There's a size limit on how much genes we can give people extra and so forth. So, I think it's probably not something that we can address right now, but I think the path is what I described.
Thank you. Another comment, excellent presentation. You touched on X-linked chromosome syndromes, and I noticed ARX on your graph. Can you comment on precision medicine for ATRX syndrome related epilepsy?
Yeah, yes, you're correct. ARX is one of the conditions. I don't know of any developing gene therapies for ATRX or ARX mediated epilepsy at this point. I think a lot of these research efforts and gene therapies are driven by parent organizations and by pharmaceutical companies. And for part of that, there needs to be kind of a threshold of how much information we have. So the rarer the condition and the less we know about it, the harder it is to get towards that. And so it may not have reached kind of critical mass at this point to get a therapy off the ground. But I think when we start to get these more commonplace, they may be easier to apply.
Where do you recommend for gene testing?
So I think first is to start with your doctor and ask about it. There are lots of different companies that perform lots of different types of tests. And so it's not just a matter of saying, I have epilepsy, I need genetic testing. There are at least, 15 to 20 different types of testing for epilepsy genetics. And so it really needs to be a discussion about, when did the seizures start? What kind of seizures are they? Are they associated with a brain malformation? Are they associated with other neurologic symptoms? What testing has already been done in order to select the appropriate test for you? And then there are a lot of private companies that do testing. You want to make sure you use a reputable company, and hopefully the doctor's office would have chosen that based on their experience. And so I think that's probably the best place to start.
Thank you. I think that's all the questions in the chat. We have 1 or 2 minutes left. Are there any other questions for Dr. Park before we end this session? I'm not hearing anything, and I don't see anything else in the chat. So we will say thank you so much again, Dr. Park. That was very informative, so interesting. We really appreciate your time and sharing your considerable expertise with us. That was just terrific. So thank you so much. We have
Thank you.
1 minute before our next presentation. So Dr. Park, we appreciate your time, and we will begin again in just 1 minute. So thank you all for being here, and we will begin again very shortly.